The other means by which plants can overcome an iron deficiency symptom is via strategy 2. And strategy 2 occurs in grasses. So how does this work? This is different than strategy 1. So strategy 2 is simply the release of a compound or compounds called phytosiderophores. Some people refer to these as mugenic acids. I like just saying phytosiderophores because it's a catch-all term. This is what grasses do when they perceive an iron deficiency symptom. Within the cytoplasm itself, there are enzymes that create the phytosiderophore, all right? Nicotinamine, which is a precursor for mugenic acids or phytosiderophores. And maybe a simpler way of thinking of this is that within the cytoplasm, there's a phytosiderophore that's created when an iron deficiency is perceived. And there's an actual pumping mechanism to pump this compound, this phytosiderophore, across the plasma membrane. The phytosiderophore actually acts like a chelate, and it can go out into the rhizosphere and find iron and complex it and carry it back to the root itself, right? So there's one thing that's different between strategy 2 versus strategy 1. Strategy 1 does not create phytosiderophores or chelates inside the plant root itself. The second part that's different in strategy 2 versus strategy 1 is the fact that the complex itself is completely pumped across the plasma membrane. And this is different than strategy 1, whereby a chelate would carry iron 3 or iron 2 to the outside of the root and then release it so iron 2 or iron 3 could readily be transported across the plasma membrane. In strategy 2, the entire complex is carried across the membrane itself. So here's some interesting data that tells you that this occurs across a wide range of grass species, and that phytosiderophores, or the creation and release of phytosiderophores, are induced by low nutrient, or in fact, in this case, low iron conditions. So what this table shows you, I recreated this table from some pretty old data. It's not old to me, but it might be old to some of you on this call. This plant species that Bromheld and Marshner looked at were barley, wheat, oat, corn, and sorghum, all grass species. And they looked at the concentration of phytosiderophores that were generated from these roots when the iron concentrations that the roots were seeing were great. And you could see that there were some phytosiderophore concentrations. This is in, I don't know if you really care to know the units, micromoles per gram of root per day, but not much. But when iron was absent, the concentrations of phytosiderophores that were created increased tremendously. And this was some early work that helped to prove that phytosiderophores were created by plant roots themselves when the plants themselves were perceiving an iron deficiency.